Okay, so the question is about how to calculate genetic distance. Okay, so, by the way, are you interested in watching an example of calculating genetic distance?
Yeah.
Because if you are, come on in, let me grab one more chair. There you go. Well, maybe there you go. There we go.
Thank you.
Okay. So if we're going to calculate genetic distance, we have to have a cross. So what genes should we use? I just need some letters, numbers. We make it easy and use gene A and gene B.
Yeah.
Okay. Is that okay? Okay. So the goal is to make a dihybrid. So if we're using A's and B's, what's the genotype of the F1 dihybrid? It's going to be big A, little a,
Big B.
big B, little b. So there's a dihybrid. So now that's essential. Oh, and by the way, if we're going to do genetic mapping, we're going to cross the dihybrid to a tester. So here's a dihybrid. So we have to cross the dihybrid to a tester. Okay, so what's the tester's genotype going to be? Homozygous recessive. And in this case, we do the normal assumption that capital letters are dominant and lowercase letters are recessive. So the tester genotype would be little a over little a, little b over little b. Oh, and by the way, I shouldn't be using semicolons. What should I be using? I should be using, if we don't know, so the question we're asking is, are these genes linked or not? So if we don't know, that's when you use the dot. So we're trying to figure out, are A and B on the same chromosome or not? So we're not sure yet, so we don't use a semicolon, we just put a dot. Okay. Please, of course.
For this one, can it be like A capital over the A capital?
No. So the definition, right, so good question. The definition of the dihybrid is that it's heterozygous at every gene or locus that we're talking about. Good question. And we're going to look at the F2 organisms that are produced. And that's where we're actually going to get the data we use to calculate genetic distance between A and B. First, we need to do parental haplotypes and recombinant haplotypes. We need to figure out which haplotypes of gametes are going to be recombinant types and which are parental types. In order to figure that out, we need to know what cross produced the F1 dihybrid. So now I need you to help me figure out what were the genotypes of two parents that were crossed together to make an individual that's got that genotype.
So, big A, big B.
So you want this, yeah, this individual could have big A over big A. We're talking about diploids.
Big B over.
Okay. So if that parent's got all the capital letters, then what's the genotype of the other parent? It's got to have all the lowercase letters. The reason I was asking you to do this for me is because these are not the only, this is advanced genetic mapping, so feel free not to pay attention if you don't want to. But at some point you will see on an exam, if not sooner, that these are not the only genotypes that you could cross together to produce the F1 dihybrid. So just we'll use the ones that are written in black here. But for example, I just want to show you dots, not semicolons. So if we have one parent that's homozygous dominant A and homozygous recessive B, what's the genotype of the other parent that would still produce this dihybrid? Right. So this individual would have the lowercase a's and, uppercase b's in that parent. So either of those crosses, the black one or the blue one, produces that F1 dihybrid. The reason this is critical is because your parental haplotypes are dictated by the genotypes of the parents. So the black parent here, big A over big A, big B over big B, would produce all gametes that have big A with big B. And if that was crossed to the recessive parent, that one would have haplotypes that are little a, little b. So those would be the parental types for the black cross. What would it be for the blue cross? Right. So here, everything we did today in class, every time you saw a capital letter with a small letter, it was recombinant. You have to know what that cross is that produced the F1, because that's what tells you which are the recombinant types.
So the blue one is a recombinant.
So the blue haplotypes that are circled there are the gametes that would be produced by the blue genotype parents. So the only point is you can't assume that the parental types are always the capital letters together and the lowercase letters together. Every question you do, you have to look at the P0 generation and for yourself figure out what are the parental haplotypes. For the rest of this, we'll just stick with the black cross. Okay. So we have big A with big B as a parental type, P, and the little a with the little b as a parental type.
And the blue one also parental type, but we're not going to use it.
That's right. So those would be parental types, but we're not going to focus on those for this example. Okay. So, F2s. We need to write out the genotypes that are possible for the F2s.
All have lowercase a and lowercase b.
So every one of them is going to have a slash lowercase a because that came from the tester. Right? The tester gametes are all little a with little b. So every single F2 is going to have a little a and a little b. And there are 4 different lines because I know there are 4 different combinations of capital A and small a and capital B and small b. So you can have the big A's with the big B's, big A's with little b's, little a's with big B's, and little a's with little b's. And those all have dots in between them. Okay. Right. So now what you're trying to do is reverse all that hard work I just did writing out the genotypes, which is, yes, how you know the genotypes of the gametes from this dihybrid is the way I've drawn the genotypes here. It contributes, right? Every one of those gametes that are produced from the dihybrid, they're haploid gametes. They've got one copy of gene A and one copy of gene B. So you either get big A with big B, or big A with little b, or little a with big B, or little a with little b. That's Mendelian. You get all possible combinations in equal frequencies. So that's a 1 to 1 to 1 to 1 ratio. You should have expected equal numbers of those 4 different types if they're unlinked. which means independent assortment is happening. So if A and B are on different chromosomes, you should have equal numbers of the 4 of these. Okay. Here's where things get really tricky for me. It's easy for the rest of you. We have to fill in how many F2s of these 4 different genotypes we observed. Do you want to make up some numbers? Do you want me to make up some numbers? So if you want to make them all equal, so we can start with that. So if we observe 1,000 total F2s, and there are 250 of each of the 4 different genotypes, what's the genetic distance between A and B? Where are the recombinant haplotypes? So we're going with the black.
So A capital and B small. The second one.
So this is, right, so the big A with a little b is not something that was in a gamete of the P0 generation. So that one is recombinant. And the little a with the big B is also not a parental type. So that's also a recombinant type. So to calculate genetic distance, it's the fraction of recombinants, which in this case there are 500, or 250 of the first, 250 of the second, divided by the total number of F2s that you're studying. So that's how we calculated a recombination fraction of 0.5. You multiply it by 100, then you get centimorgans, 50 centimorgans.
It's always 50?
In class, it's always 50 when the genes are unlinked. In reality, you never, if you're actually studying real organisms, you never get exactly the same number of each. So you might calculate 49.8 centimorgans or 47.2 centimorgans, something close to 50. It would actually be very rare. Unless you study an infinite number of F2s, then you'll get some fractions very close to 0.5. Rarely exactly 0.5. Now, let me make up some F2s. So we're going to assume it's the same. We're going to assume it's the same cross design. So P generation is capital A's and capital B's on one parent, small a's and small b's in the other. Make a dihybrid that way. You cross it to a tester, all lowercase letters, and that produces our F2s. So we get some capital A over small a, we get some lowercase a over lowercase a, and that means that one has to be big B over little b over little b, big B over little b over little b, I think, if I did that right. So we've got a big A, big B, a big A, little b, a little a, big B, and a little a, little b. So once we cross out the haplotypes that came from the tester, then we have the 4 different possible haplotypes that would come from meiosis from the dihybrid. Okay. Now this is going to stretch my math skills. Warm up a little bit. So let's say we've got, I'm going to try to make it total 1,000. This is the part that's hard. Okay, so if I have 390 of those, and if I have 420 of those, that's 810. maybe that totals 1,000. Let's see. 525, 530, 610, 700, 1,000. Yeah. So what's the genetic distance between genes A and B?
Is it between big A, big B, or is it just recombinant? It's already recombinant.
So we just want to know how many gametes were recombinants. So we have to figure out of these 4 types, big A with big B and so on, which 2 of those 4 are the recombinant types. I told you this is using the exact same cross that we used on the previous slide. So the parental types are big A with big B and little a with little b. So I just go down and I label which ones are P, parental, and which ones are R, recombinant. That must be parental. Okay. So how many recombinant F2s did we observe?
So we add them in bottom by 1,000.
Right, so whatever that is, 190 over 1,000, 0.19 or 19 centimorgans. 0.19 would be the recombination fraction RF.
We convert it to centimeters, how do you do it?
So centimorgans.
Yeah, if you gotta convert it to centimeters.
So you can't convert, that's a great question. So you don't, I wasn't sure if I should have mentioned centimeters in class because I was worried that I might confuse things more than necessary. Centimorgans is measurement of genetic distance. Centimeters we never use in genetics to talk about physical distance. So there is actually no way, well, in our class, there's no way to convert between genetic distance and physical distance. So
We can convert it to MU.
you can convert genetic distances to other measurements of genetic distance. So you can convert recombination fraction, map units, and centimorgans. That's all genetic distance. But you can't, there's no formula for changing a genetic distance into a physical distance. So, and that's one of the slides I skipped in today's class, which, like I said, maybe I'll come back to at some point because I think it's a very interesting concept, but what I think is interesting doesn't mean that you think it's interesting. So there's no way to convert genetic distance like centimorgans. There is no formula to say if 2 genes are 10 centimorgans apart, they are some number of base pairs apart. That would be the case. Okay. So here's a graph of physical distance, base pairs on the y-axis, and genetic distance between 2 genes, centimorgans, on the x-axis. Now it would be really nice if there was a linear relationship so that you knew that if 2 genes were 5 centimorgans apart, they were, I don't know, 100,000 base pairs apart. That would be really convenient if there was an absolute straight line and you knew that if you knew the genetic distance, there was some sort of, it doesn't even have to be linear, it could be a curve. If there was some function where you could say I take a genetic distance, I convert it to physical distance. It's not true. It doesn't, for a lot of organisms, there is no simple relationship between how physically, which is strange. There is not a simple relationship between how physically far apart 2 genes are and how likely it is that recombination happens between them. And if you go back and look at the slide that I skipped in class today on that topic, you'll see this plot for a human chromosome, and there's absolutely no relationship between how physically distant 2 spots on the chromosome are and how frequently recombination happens. Another screen. So conversion of centimorgans to map units. So if you have 10 centimorgans equals how many map units? They're the same. So the calculation of moving from recombination fraction, which is always our initial calculation, how many recombinants divided by how many total F2s. So if the recombination fraction is 0.1, then you either multiply, it's the same thing. You multiply it by 100, that gives you map units. And multiplying by 100 also, and this is why I apologized, because it makes no sense. It's historical, the reason there's so many different terms. It's not that geneticists sat down and thought, you know what, we should make things really confusing. Let's use a bunch of different terms.
It's not the same thing.
So they're the same, yeah, so map unit, one map unit is the same genetic distance as one centimorgan.
Is there a difference between them?
None at all. Absolutely none. They're exactly the same thing. There's no reason to use one instead of the other. They're absolutely identical in every way. They just have different names. So now you really understand why I'm apologizing, because I just wanted to talk about this in class because it seems to be, every textbook in genetics talks about all of these terms. And I just wanted to be really explicit and point out, I acknowledge, you'll see some questions that talk about centimorgans, map units, and recombination fraction. So it's important to be able to convert them. Fortunately, it's pretty easy to do.